Okay, 200 nanometer. Okay, so that is the size of human DNA inside the chromosomes is just 200 nanometer. Okay, move. So let's now focus on the purines or on the nitrogenous bases. So in the purines, we have adenine and guanine in the double ring or in the DNA, and in the pyrimidine, we have thymine, cytosine in a single ring. Okay. Next slide. The bases always pair up in the same way, a purine with a pyrimidine. So as I've said, there is no such thing as a adenine will be pairing into guanine. No. So always remember that it is constant in the DNA and RNA that in a purine, adenine forms a bond with the thymine and cytosine forms a bond with the guanine. So this is actually the illustration of it, just to be clarified, just to give you a illustration of the nitrogenous bases. Next slide. Okay, let's now discuss the replication of DNA. Basically, there are steps in DNA replication. So the first step is DNA must unwind and break by the hydrogen bonds. So basically, if the DNA will break with the hydrogen bonds, so there is a DNA replication. And second with that, we have each strand is used as a template or a blueprint of the original DNA. So that is why there is a DNA replication, because it is used as a DNA, or the original DNA is used as a template of the blueprint. Okay. Third is a two new strands of DNA are formed from the original strand by the enzyme DNA polymerase. So basically, in the DNA replication, it will be making or forming another strand based on the original strand of the DNA by the use of an enzyme and DNA polymerase. Okay, moving on. We have it now. Diyan papasok si transcription or how RNA is made of. As I've said, DNA will be ongoing on the DNA replication, and that's the time that the process of transcription can make the RNA or the ribonucleic acid. Okay, basically it said that just a DNA polymerase makes a new DNA. Yes, a similar enzyme called RNA polymerase makes a new RNA in the process of DNA replication. RNA polymerase temporarily separates the strands of the small section of the DNA molecule. This exposes some of the bases of the DNA molecule. This is actually in the process of transcription. Along in the one strand, the RNA polymerase binds complementarily in the RNA nucleotides to expose the DNA bases. That is now the process in transcription, where the thymine now will be replacing the nitrogenous bases called uracil. Okay, moving on to the next slide. It said also that the RNA polymerase moves along and makes a strand of a messenger, which is the type of an RNA. Okay, it codes as a messenger RNA because it carries DNA message out of the nucleus and into the cytoplasm. And mRNA is a single-stranded. So later part, we will be digging deep with this 3 division of the RNA. So when the RNA polymerase is done reading the gene in the DNA, it leaves. Yes. And it will leaves, the separation of the DNA strand will reconnect and be ready to be read again when it necessary. So the messenger RNA moves out of the nucleus and finds ribosome. So on the ribosome, amino acids are assembled to form proteins in the process called translation. So always remember that transcription is always reconnect to the process translation by the assembling of amino acid to form protein synthesis, and that is called as the process of translation. Moving on, so now let's discuss ribonucleic acid or the RNA. So basically, ribonucleic acid is usually called as an RNA. It is a biologically important type of molecule that consists of a long chain of nucleotide units. So the differences between the DNA and RNA is in the nitrogenous bases and of course in the structure of it, because in DNA it has a double helix bond or a double helix structure. In the RNA we have the single non-helical structure. So continuing, it is a single-stranded chain of nucleotides that contains genetic information or genetic materials and functions for the synthesis of proteins and also to transfer genetic information from one generation to the next. So that is the function of RNA. So basically there are three types or three divisions of RNA in a cell. We have ribosomal RNA or the rRNA, the messenger RNA or the mRNA, and the transfer RNA or the tRNA. So then the main function of it is to make proteins after taking instruction from the DNA. They are temporarily present in the cell. So moving on. So this is the summarization. This is a very good summarization of the division of the RNA. So basically RNA can be a messenger RNA, ribosomal RNA, and transfer RNA. RNA can be messenger RNA in a sense that it carries instructions and carries instruction from DNA to ribosome. So that is the reason why RNA can be messenger RNA or also called mRNA, which function is to carry instruction from DNA to ribosome. So RNA can also be a ribosomal RNA or also called rRNA. So which function is to combine with the proteins to make up a ribosome. Aside from that, RNA can be a transfer RNA. Transfer RNA is also called as tRNA, which function is to bring amino acid to ribosome. So always remember this, the simple summarization that make you explain or discuss the division of RNA. Why RNA can be a mRNA, rRNA, and tRNA. Okay, continuing. So let's discuss what is mRNA or the messenger RNA. Basically, it also known as mRNA. Messenger RNA is a single long chain of nucleotides. It is a molecule of RNA encoding a chemical blueprint for a protein production. mRNA is transcribed from a DNA template and carries coding information to the sites of the protein synthesis that make up with the ribosome. So always remember that messenger RNA is a carrier of coding information to the site of the protein synthesis. That coding information is from the DNA template itself. So that is the function of mRNA or messenger RNA. Next to that is mRNA as a DNA genetic information is encoded in the sequence of nucleotides arranged into codons consisting of 3 bases each, and each codon encodes for a specific amino acid except the stop codons that terminate protein synthesis. So codons basically is a code which is a component of a DNA template that when the messenger will carries this one, it will carries a coding information, and that coding information is from the DNA template itself. Okay, continuing our discussion, let's move on to the ribosomal RNA. Ribosomal RNA, it is also known as rRNA. Ribosomal RNA is the central component of the ribosome, the protein manufacturing machinery of all living cells. So basically, the formation of ribosome is in the ribosomal RNA. Next, ribosomal RNA continued, or the continuation of it, it has 2 units. We have one large and the other small. So, ribosomal RNA, the illustration is this. So, as I've said, it has 2 units. We have the large subunit, or this one, and the small subunit, or this one. Okay, so always remember that one. Next, so basically the function of it is to provide a mechanism for decoding. If messenger RNA is for coding information, rRNA or the ribosomal RNA is for the decoding decoding the mRNA into amino acid and interact with the tRNAs during translation. So that is the function of the ribosomal RNA. Always remember that one, na if mRNA is for the carrier of coding information, the rRNA now will decode the coding information into amino acid and interact it to the transfer RNA during the translation process. So the tRNA then bring the necessary amino acid corresponding to the appropriate mRNA codon. So always remember that one. What is the connection between the mRNA, ribosomal RNA, and the tRNA? Always remember that messenger RNA will carry the coding information. If the messenger RNA carries the coding information, the rRNA now will decode the coding information or the information. It will decode the information. After that one, after decoding it into amino acid, it will interact to the transfer RNA or in the process translation. So always remember the interaction or the connection of the three division of RNA. Next is, this is actually the codon match up with the anticodon to create protein. So as we can see, we have codon 1, we have guanine and cytosine and uracil. Codon 2, we have adenine, cytosine and guanine. Codon 3, we have guanine, adenine and guanine. Codon 4, we have cytosine, uracil. And codon 5, we have cytosine, guanine. And for the codon 6, we have adenine, guanine, cytosine, and cytosine. And for the codon 7, we have uracil, adenine, guanine. So that is actually the codon match of the RNA. Next. So basically, the conclusion of this topic is the nucleic acid or the multi-coded complex molecule in the biological system from virus to human. So nucleic acid is very important. Why? Because nucleic acid carries the genetic information by the means of DNA and replicate it by the means of RNA. So that is very important. Why? Because that both of process will actually carry a gamete, and that gamete will complete the organism, or you as a human or animal or animals and all the living organisms. So that is the importance or the conclusion of this topic. Aside from that, exploring the nature properties possible to understand the molecules and to bring the desirable modification in the future world for the benefits of living system. So by knowing the nucleic acid, you will know where does the organism come from, what are the systems inside, or what are the process inside the living organism, which is basically one of the process is the nucleic acid by the means of the DNA and RNA. Aside from that, so that will end our discussion for today's session. So for the topic nucleic acid, so that is the important thing or the conclusion of everything. Why do you need to study these macromolecules, which is called nucleic acid? Because it is very important to explore the nature and properties inside the living organism, and aside from that, to know what are those processes inside your body. Okay, so thank you very much. If you have questions and clarification, you can raise that in our group chat. If none, then thank you. Okay, bye.